Thank you, John, for the introduction, and thank you all so much for coming. It means a tremendous amount. This is my thesis defense. The title of my thesis is Magnetogenetics, Remote Control of Cellular Signaling with Magnetic Fields. A quick overview of my talk. I want to kind of motivate the work I'm going to be talking about, and that is artificially affecting cellular function through three specific aims. The first, remotely actuating TRPV1 ion channel using a tethered nanoparticle and applied magnetic field. In specific aim 2, I'll discuss a bit about the mechanism by which magnetic fields can interact with iron oxide nanoparticles to cause ion channel opening. And then in specific aim 3, I'll discuss development of low power, low cost magnetic stimulation devices to power the technology that we've developed. And then in the end, conclude with some proposed future. When we think about how to affect cell fate, this is really the question of modern pharmacology. If you're in a disease state, what can you take, what can you put in your body to interact with your biology to change something? This is initially realized as small molecule drugs like aspirin, that sort of drug, where somewhat serendipitously, medicinal chemists can find a chemical in nature that will interact with the body and treat some disease. Moving along from that, starting with recombinant insulin, we went to protein drugs. Protein drugs now can more intimately associate with biological functions and work with more complex pathways. What I want to talk a bit about today is this idea of a nanodrug. So this is a supramolecular approach where we're marrying material science with medicinal chemistry. And there's a number of ways you can use nanomaterials to drug a target. There's the most common one is probably drug delivery. And this is where some sort of nano size, and here by nano I mean very small. So this is 10 to the negative 9th meters, people that don't know. And that's on the size of proteins. So these particles can envelop drugs or proteins and enable their effective delivery into a cell. They can be a diagnostic, so they could be a contrast agent for brain imaging or cancer imaging or that sort of thing. And what I want to highlight is the idea of a nanoparticle as an actuator. So what is that? A nanoparticle actuator works on a cell in an orthogonal way. And what that means is we want to be able to stimulate the cell in a unique way so that it won't stimulate any other target. And so to do this, we use a nanomaterial transducer. So kind of the model I'm proposing for this nanoparticle actuator is you have some target, some cellular component that you can attach a nanomaterial to. This nanomaterial will translate an otherwise orthogonal or otherwise a stimulus that otherwise wouldn't affect the biology for some desired outcome of altered cellular activity. So this could be signaling, gene transcription, cell fate, that sort of thing. The relevant type of remote activation that I'm going to be talking about is what we call magnetogenetics. And that's the use of magnetic fields coupled with a magnetic material such as a magnetic nanoparticle to affect cellular behavior. And this can be done to apply forces to the cell membrane, to twist, to pull, to let ions through, to couple receptors. And this has the benefit that magnetic fields freely penetrate tissue. This is in direct analogy to optogenetics, which some of you may have heard of. And in optogenetics, light-sensitive ion channels can be remotely actuated with a specific wavelength of light. This, however, suffers from the fundamental limitation that light of this wavelength cannot penetrate more than 1 millimeter into the skin. So we want to propose this as a more clinically applicable optogenetics. A bit about magnets and magnetic fields. So our stimulus is magnetic fields, and the nanomaterial is iron oxide nanoparticles on the order of anywhere from 10 to 40 nanometers. Magnetic fields can really be static or oscillating. So oscillating, if you've heard of radio frequency, like what powers your phone when you make a phone call, is really nothing more than a rapidly switching magnetic field. And a nanoparticle can move around to follow the field, or it can heat up. Whereas in a static magnetic field, like your fridge magnet, it can really just apply a translational force to the particle. At this point, you might be asking yourself, magnets, how do they work? So a couple of fundamental sources of magnetism. So there are two. One is permanent magnetism, like in your fridge magnet. And this is caused by unpaired valence band electrons that can couple. And so this allows certain materials like iron, nickel, to be able to sustain a magnetic moment. Or electromagnetism. if you have current flowing in a wire, it creates a magnetic field around it so that you can have an electrically inducible magnetic field. Without going into too much depth, I just would like to discuss the types of magnetism that are relative to this discussion. So a diamagnet, things like your body, does not interact with magnetic field. So that's why a radio frequency wave can freely penetrate your body and go to your cell. A ferromagnet is a multicrystalline particle that's able to sustain a magnetic field. And that's why your fridge magnet is able to stick to your fridge even though you're not applying any magnetic field to it. And finally, if a ferromagnet gets sufficiently small, it'll become a single crystal. And this single crystal can have its moment flip thermally, it'll align in the magnetic field, but then its moment will go away once the applied magnetic field goes away. For our target, we have an ion channel that's expressed in the cell membrane called Transient Receptor Potential Vanilloid, or TRPV1 ion channel. It's a fantastic protein. It's well characterized, well studied. First discovered in '95, there's been over 1,000 papers on it. Expressed in the membrane, and it's polymodal. And what polymodal means is that many different things can activate it. So it's canonically the capsaicin receptor. So if you like spicy food, that's your TRPV1 ion channel your tongue will open. Moderate heat, so if you touch a hot object over 42°C, this will open. Strongly depolarizing voltage, acidic pH, and mechanical strain. Fairly moderate mechanical strain, and this will be important later. And it conducts the calcium ion. In 2012, our group in collaboration with Rockefeller University, the Friedman Lab at Rockefeller University, developed this system where an externally tethered magnetic particle is tethered to the TRPV1 ion channel, and in the presence of a 465 kilohertz RF field, can be gated, allow calcium in, and promote transcription of a calcium-sensitive gene construct. The RF imparts to the particle energy, which is then transduced to the channel in the form of heat and torque. And the mechanism proposed in the paper was thermal, and we're going to kind of go into what may be at play there. So in specific aim 1, I have developed a remote static magnetic activation of TRPV1 to test in vivo for therapeutic applications. And we've termed this technology nanoparticle-induced cellular regulation, or NICE. It's always important to have a good acronym. This work has led to a recent publication in Nature Medicine in January, and a provisional patent that we are currently seeking. So why use a static magnet to activate TRPV1? So the current technique requires a high-power RF radiation by using an inductive heater. So inductive heaters are large coils with huge power demands that are usually used for heating metal parts for welding and that sort of thing. High energy and capital cost, risk of peripheral tissue damage through non-specific heating, and it's not portable. This thing weighs like over 100 pounds. So in order to have facile translation of this technology, the ability to actuate TRPV1 with a static magnet is paramount. So in order to test this, I developed and used a technique that's been reported in literature called magnetic pulling cytometry. And this, much like our current system, couples a magnetic bead to the outside of the TRPV1 ion channel using antibody-antigen interaction. And then by applying a localized magnetic field, we can monitor the calcium response to that magnetic field. I constructed a probe solenoid microneedle. So this is a highly conductive core wrapped about 1,200 times by hand with Sandra Sanchez. It was wonderful. And when current is put across this coil, it creates a very strong magnetic field just at the tip of the magnet. And this allows us to interrogate very small populations of cells and look at their calcium signals. I wanted to calibrate this just to get an idea of what kind of force I could put on these particles. So to open TRPV1, I need about 1 piconewton of force. Here I used 1 micron beads, and by pulling them through glycerol, you can track their velocity, and that velocity is proportional to a force. And so we're able to map force to distance from probe. So if 1 piconewton of force gates the ion channel, we have a large dynamic range by which we can remotely apply force and see calcium signaling. This is what the assay looks like in a cartoon form. It's called fluorophore. It's a calcium-sensitive dye that, upon binding calcium, increases its 488 fluorescence. So we transfect in our ion channel, put in the dye, add a particle, some kind of magnetic field. Field acts on the particle, calcium enters and the cell turns green. So we do a lot of looking at green cells. Just a note on calcium signaling. It's an incredibly complex intracellular process that's highly regulated throughout the cytoplasm and organelles of the cell. When calcium first enters the cell through TRPV1 after it's open, a small amount of calcium enters and triggers two endoplasmic reticulum channels. These then allow a large store of calcium to be dumped into the cytoplasm, acting as an amplification of that initial calcium signal. At the transcriptional level, throughout the entire body, there's three main signaling pathways that take advantage of calcium to cause gene expression. The serum response element, the cyclic AMP response element, and the nuclear factor of activated T cells response element. In subsequent slides, we will use this along with a minimal promoter to induce calcium-sensitive gene transcription. So we have TRPV1 in a constitutive CMV plasmid. Our model system is HEK293 T cells using transient transcription. We transfect the cells, and then we first wanted to make sure the system works. So I mentioned earlier that TRPV1 is sensitive to acidic conditions, and indeed we see cells that are stained with fluorophore. So this is the background signal from just the background amount of calcium in the cell. And upon the addition of acidic buffer, TRPV1 opens and you see an increase in fluorescence, and that's a channel opening event. We then wanted to do this using magnetic pulling cytometry, and indeed it does work. Cells are decorated with antibody localizing particles to localize to the channel, and then upon stimulation with a magnet, we see increase in cellular calcium via the increase in fluorescence. We can look at it just a representative trace of an activated cell, and we see cells that don't respond just sort of stay flat, and then cells that do respond, we can see something of a biphasic response where calcium enters, gets amplified, and is ultimately sponged up by ubiquitous calcium-binding proteins in the cytoplasm. So this shows that TRPV1 can be remotely actuated by simply a static magnetic field. So now we wanted to do something with this. So we wanted to remotely actuate TRPV1 for a therapeutic effect. So to do this, in the 2012 science paper, external nanoparticles were used, which, while they were efficient, required injection of nanoparticles to the site of action. With collaborators at Rockefeller, we used an entirely genetically encoded process. So you all have magnetic particles floating around in you right now called ferritin. And ferritin is an iron-binding protein that sequesters reduced iron, oxidizes it, and stores it so it prevents oxidative damage to your cell. There's 24 individual ferritin proteins that make a protein cage to make a approximately 10 nanometer superparamagnetic nanoparticle. So now that we have this, we want to see where can we put this in a cell to make it most efficient. We can put it in the cell, just have it floating around in the cell. We can meristylate it so it'll attach to the cell membrane. Or we can attach it to the ion channel using GFP fusion ferritin with an N-terminal anti-GFP nanobody, so that upon expression, our ferritin will only be localized directly to the channel. And we wanted to test this both with the RF. I mentioned this is not portable. This is very large and takes about 230 volts to power, or a simple bar magnet you buy from the hardware store. And so again, this looks familiar. Put in the channel, the particle is now intracellular in a magnetic field. Particle is actuated, calcium comes in and promotes the expression of secreted insulin. Perhaps unsurprising, the closer the stimulus is to your sensor, the better your response. When the ferritin is directly attached to the TRPV1, we see a large significant increase in secreted calcium. And then attached to the membrane and just randomly in the cytoplasm, we see less expression of insulin, likely due to the poor coupling of our stimulus with our target. So what can we do with this in vivo? So we use black 6 mice that are adenovirally introduced with a nicer system. And that includes the ion channel, the engineered ferritin particle, and the calcium-sensitive insulin gene. When these animals are treated with RF, we see a large significant increase in plasma insulin showing that we're remotely promoting the expression of insulin. And we likewise see the same thing with a magnet. Well, while significant less. Insulin is a nice reporter because it has a physiologically relevant action, and that is reducing blood glucose. So we can also study the blood glucose of these animals. And we see here the animals are anesthetized, treated with a magnet, treated with an RF or a magnet, and have the blood glucose tracked over time. First thing to note is whenever you or an animal goes under anesthesia, your blood glucose goes up. And this is a stress response well characterized in the literature. Your cortisol, your stress hormone goes up and causes release of glucose. So that's why this is net positive. However, in the presence of RF, that positive response is entirely abrogated, and with a magnet, it's largely and significantly abrogated. And we can see over here, with treated localized ferritin with RF, we see a dramatic decrease in blood glucose, and with a magnet, a slightly less, albeit still significant. So to conclude from this, TRPV1 can be mechanically actuated simply with direct force from a static magnet. It can also be done with RF, as been shown before. As was noticeable from the data, RF is more efficient than static magnet. We have some hypotheses why this might be. It's well known that ion channels can respond to a directionality of force, although what that directionality is poorly understood. A particle in RF is able to jumble at 500,000 times per second, so it can just sample more torque space than a simple one-directional attraction to a static magnet. And finally, this remote activation scheme can produce therapeutic effects in vivo. Specific aim 2, we wanted to delve a little bit into how can RF affect this nanoparticle to affect the ion channel. So there's some standing controversy in literature about if you treat a concentrated solution of nanoparticles with radio frequency, it heats robustly, and that's readily observable. But what's happening at the nano level? Are these particles heating intensely, or is that heat diffusing away rapidly? And there's a number of papers with elegant titles such as magnetic nanoparticles heaters kill without a perceptible temperature rise. So there's obviously some confusion there. A little bit on how cells can transduce magnetic energy. Based on the size, they'll try to align with an opposed magnetic field. A larger particle, the entire particle is going to rotate to align with a applied magnetic field because that's energetically favored. And this generates torque in the particle. As the particle gets smaller, it's now easier for the magnetic moment to turn within the particle to align with the field. And this generates heat via something like a viscous loss. Some very nice modeling work done by Paolo Kablinski out of the Materials Research Center did some calculations as far as how can we explain this bulk heating versus this bulk heating effect, whereas on the nanoscale we don't believe we can be sustaining these temperatures. If we look at a concentrated solution of nanoparticles, we indeed see rapid and robust heating in the presence of RF. And if you'll excuse the equations, this is basically a summation of the point heat sources of each particle. So each particle is going to heat, that heat's going to diffuse away and contribute to a bulk heating, but at the single particle level, diffusion is going to dominate. And so thermal diffusion is going to rapidly wick away produced heat. So how can we sort of experimentally test this to add our 2 cents to the literature on this? So I wanted, in the 2012 science paper, we used a 20 nanometer iron oxide particle, which is right at the cusp of that brown and needle relaxation transition. So it heats robustly and it also applies torque. If we go to a larger particle, it does not heat very well in this kind of RF, but it does apply a substantial torque. So this could potentially give us some light on, is this a mechanical method or a thermal method? As was shown, and as I said, particles, as they increase in size, decrease in amount of temperature increase will be evolved upon treatment. And the 40 nanometer is not shown here, and indeed, the 40 nanometer particle shows this trend with almost negligible heating over the time scale of the experiment. So I wanted to test our TRPV1 expressing cells with these non-heating 40 nanometer particles and see if they'd be able to actuate TRPV1. And so what we can see is that they are indeed. Just to go over a little bit of how they can activate. Here, I call this a complete activation. So this is the ion channel opens, the ER is able to propagate that signal, and you see a large calcium increase, which subsequently tapers off to the sponging up of calcium binding proteins. One thing that's interesting to note here, the RF was turned on at about 10 seconds. So there's a huge delay here. And so we think, again, that has something to do with that random directionality, where the RF is not able to activate the cells until it gets that correct directionality. We can also have a partial activation where ion channels keep opening, but it's not enough to stimulate the ER to amplify the signal. And then finally, stable means that it expresses TRPV1. These are stable cell line. Blank does not express TRPV1. And in blank cells, you can see no response or a slight increase in fluorescence. This is likely due to aggregates bumping into the cell. And when we analyze 31 cells, so this is a manual analysis, so you can think of it somewhat qualitative. 22 of the 31 TRPV1 cells treated had greater than some threshold increase, showing an effective calcium signaling, where only 2 of the 31 without TRPV1 show increase. So this preliminary data sort of tells us that this is working and something's going on in the presence of TRPV1. So now can we do this with our transcriptional reporter? So we used our proinsulin readout, calcium-sensitive insulin, treated the cells, and again we see a significant doubling in proinsulin as opposed to control, showing that we are effectively actuating this actually as well as the 20 nanometer particle. In the original paper, they saw twofold enhancement of plasma secretion, proinsulin secretion. And then finally, ongoing work to be finished in the very near future. We want to test on ion channels with different temperature sensitivities. So TRPV1 is fairly sensitive to temperature. What if we tried a channel that was less or not at all sensitive to temperature? And if it were able to be opened, we would be quite confident that it's an athermal mechanism. So TRPV2 is a related ion channel that's only activated at extremely noxious temperatures. And I've cloned a mutant of TRPV1, where by modifying the pore region, it completely abrogates the heat sensitivity. And those experiments are ongoing. Finally, in specific aim 3, I sought to develop devices by which we could inexpensively produce and distribute this technology to enable broad adoption. In order to do this, we need a quantitative readout of populational level calcium flux. So the fluorophore assay, as you saw, is nice, it gives a nice visual, but it's hard to get a truly statistical representation of what's happening. And the insulin reporter is nice, but it's expensive, and the assay is somewhat time-consuming. So our collaborators helped us clone this construct that again has the triplicate of the 3 calcium-sensitive signaling pathways, our minimal promoter, and now that 5' of a GFP gene. So that when calcium flux occurs, we can fluorescently measure it at the transcriptional level using either a plate reader or the flow cytometer. So we put the channel, put a particle, calcium comes in, GFP is expressed. We can just measure the fluorescence at 480. First, we wanted to validate the sensitivity of this reporter. So would it be able to see a single activation of a channel? So for TRPV1, and just to check also TRPV2, we added a 60-second dose of the agonist. So we would chemically open all the channels and then measure the response. And indeed, we've got quite a nice dynamic range for cells that express the channel in response to their agonist, and no background from cells just expressing the reporter, here SCN GFP. With a reporter, I wanted to make 2 apparatuses. The first is one that can do a very strong field at low frequencies, and one that can do weaker field at high frequencies. And I'll explain that. So the first one, I wanted to be able to treat at 1 hertz or longer, so 1 treatment per second or longer, at very strong field strengths, greater than 200 millitesla. Just for ballpark, an average small MRI could be 2 Tesla. So this is about 10 times weaker than MRI, which is really strong. This uses an array of permanent magnets to apply magnetic force to cells in a 96-well format to allow for high throughput screening of mutants, particles, any library that you can choose. This shows the magnetic decay around one of the permanent magnets that I used. This is a picture of the device. I was going for cheap and simple. So this is a box with a solenoid motor that lifts an array of magnets and then falls back down with gravity to a set point so that the cells will see the same magnetic field across the plate and throughout the time course of the experiment. This is controlled using an Arduino controller, so we can do any number of frequencies from DC all the way down to 1 hertz. Now we want to see if does it work. We use the 40 nanometer particles, and we indeed do see that it works an hour of treatment at 1 hertz. So this is particles and magnet, no particles and magnet, particles no magnet, and neither. And we see a significant increase in GFP signal only when there's particles present, showing that the device itself isn't affecting the cells. The device is actuating the nicer system in vitro. So I didn't talk much about the radio frequency device that we're currently using for our work. And it's an RF inductor. And this looks like 2 to 2.5 turns of thick copper wire around an air core that produces 3 main fields. It produces an axial magnetic field that's actually rather weak, about 30 millitesla. A very strong axial electric field on the order of kilovolts per meter. And so the purpose of this is these devices are usually used to heat bulk iron. And so in bulk iron, this switching electric field can cause heating much more rapidly than with a switching magnetic field. And then by Faraday's law, you have an induced magnetic field around the circumference of your device. As you can see up here, if we want to propose this as a translational device, you're never going to carry 220 volts on you, or 2 kilowatts, or 10 amps, or I would never put my arm in a kilovolt per meter. What we ideally need is we need a small, low energy, low cost device, something you could fit in your pocket, something that would be able to actuate the system in a clinically relevant setting. Perhaps something that looks like this. This is a wireless charging coil. You may have seen these used for charging your phone or charging your small devices. And these work on exactly the same principle. They oscillate an AC electric field to induce a magnetic field at the surface of the device. And actually, the magnetic field is quite strong for the powers put into it. It's about 2 millitesla at 100 kilohertz. And these are designed for efficiency. So these are designed to use low power, low electricity with minimal loss, as opposed to the RF inductor, which is intended to maximize loss. And you can see that with the power requirements. So this would be a very attractive potential translatable device. Again, does it work? We put on 20 nanometer iron oxide particles, treated for an hour, and indeed we see a significant increase in GFP expression. And this is repeatable. Again, the same controls of no magnet and no particles. So this is a significant but somewhat modest increase in GFP expression. So with these encouraging results, we're in the process, myself and Matt Breyer, working to create a larger scale and slightly higher power device that we could see better expression, as well as move into in vivo studies. So with that, just a few directions that I propose to see this work going. Collaborating with Dr. Sandra Sanchez, we're working to remotely gate ion channels using gold particles and laser light. And this has benefits of very high efficiency and might be very applicable for a number of laboratory techniques. We could also, we're remotely actuating an ion channel. So we're letting ions into the cell, and if you let positive ions into a cell, you can depolarize the cell. And that's how an action potential works. When I move my arm, an action potential is moving along a nerve to tell my muscles to move. So if we're able to remotely induce that action potential, we could control neuronal behavior. And indeed, working with our collaborators, I helped design a magnet to be able to remotely stimulate neurons. And we can see here in a patch clamp study, the presence of a magnet is able to fire a neuron. And that's working preparation. We could think of other cellular architecture to work on. So G-protein coupled receptors are the most drugged target currently. So if we were able to remotely actuate these, there is any number of medicinal applications that could be used. So just as a model, I used the angiotensin II type 1 receptor, and in the presence of a 1 hertz magnetic field, we see a very nice trend towards increased activation. And this is increased phosphorylation of ERK, without going too much into the pathway. And then finally, TRPV1 is a main mediator of a number of types of pain. If we're able to shut down TRPV1 in those particular nerve fibers, we could shut down pain transmission from site of injury to the central nervous system. We could do this by mechanically overstimulating native channels to just simply shut down the native pain response. Or our collaborators have cloned a chloride variant of TRPV1 that, when actuated, would hyperpolarize a neuron and prevent action potential from firing. And with this, we think we could develop a remote pain management scheme. So just big picture conclusions, we can gate nicer with the static magnetic field, which has tremendous clinical benefits. RF heating effects at the nanoscale are improbable, and the current data support a mechanism that may be athermal. And I've developed devices for facile magnetic treatment of cells that could be further moved into a commercial technology. None of this work would be possible by myself. I have a tremendous amount to thank to Professor Dordick, Professor Kane, who allowed me to sort of chase down the lead, fail tremendously, and maybe get something of worth at the end. My committee for their helpful input. The collaborators at Rockefeller have been absolutely instrumental in all of this work. Working with Sandra has been delightful troubleshooting experiments. Everyone in the Dordick and Kane group. I've had fabulous mentors here to teach me everything from cloning to working with cells, Western blotting. A number of colleagues and friends have just made my 5 years at RPI just fantastic. Incredible core directors and everybody that works at RPI. And finally, my wonderful family that supplied all that fantastic food beforehand. So with that, thank you so much for listening.
So are there any questions from the general audience?
So is some of the limit to how much change in expression you're getting just background expression? You need a much more tightly regulated gene construct?
Yeah, so we, as you can see in all the experiments, there's quite a background. And so we initially thought it was our gene construct. The promoter we're using is the interleukin-2 minimal promoter, which is, through the literature, quite tight. So we weren't sure that it was that. What we think it most likely is in every cell you have about 100 nanomolar calcium in your cytoplasm. And we think that's just a background level of signal that we're always going to have. So in order to get convincing results, we have to far surpass that. And likewise, in vivo, we're going to have to surpass that. Victor?
Could you comment on what further challenges are faced to get this eventually to clinical trials?
Huge and numerous.
Some of the big ones.
Some of the big ones. So, easy one, developing a device that's able to penetrate deep into tissue. The magnetic field off of this is excellent at the surface, but decays with about 1 over r squared. So if you want to treat somewhere deep in the brain, that's going to take some energy. Being able to safely and efficiently express the ferritin particle in heterogeneous tissues, that's going to be a serious issue. The FDA will probably be the largest one. Gene therapy, so this would be a gene therapy, and going into it, I thought gene therapies were nowhere close to being used. And there's actually over 100 in the clinic right now, using similar viruses and similar delivery methods to what we're using. So we think it's quite feasible, although probably not in the next year or two. Brady?
So just to follow up on that, could you tell us how the level of ferritin you guys have, like the number of particles per cell or concentration per cells, versus what would be found at a maximum in natural cells?
It's an interesting question. So ferritin is typically an extracellular protein. So you typically get circulating ferritin. Your concentration of iron in the cell normally is quite close to zero depending on the tissue that you're in. So there's some concern as far as toxicity. You know, if you're accumulating a lot of metal ions, could that cause some toxicity? In our in vitro studies and some preliminary in vivo studies, we don't see induction of apoptosis. We don't necessarily see necrotic cell death. Speaking of a key challenge moving forward, investigating that will definitely be. Jake?
Yeah, I was just interested to know, you mentioned a couple times that directionality or like the amount of force required was like 1 picoton. So how was that determined before this kind of system?
Sure. So We think about forces at the pico level, so that's 10 to the negative 12. It's very, very low. Just for some perspective, thermal motion is about a femtonewton, 1 to 10 femtonewtons. So you wouldn't want thermal motion to set off your channel, so nature sort of designed it so it's about 100-fold higher than that. Most of those force studies were using AFM to map breakage forces. So they've done this with an antibody antigen is anywhere from 50 to 100 piconewtons. Streptoabamine is closer to 200 piconewtons. Those studies used AFM to measure forces needed to open an ion channel. Prof?
If I were to introduce a small molecule or antibody in my body, it will most likely be degraded and also can be used through urine and
Yep.
so on and so forth. So likewise, if I were to inject nanoparticles in my body, what's going to be the fate of that? How do you plan to, let's say, remove it once it has provided its cellular effect?
Sure. So a couple steps to that question. If you inject nanoparticles in the body, they clear rapidly. Based on the size, order of minutes, they'll go to the kidneys, the liver, and then ultimately be excreted. That's why we wanted to get away